Can you hear me? Can you hear me?
Good morning, sir.
Good morning.
Good morning, sir.
Good morning. Ask them to join.
Okay, sir.
Yes, sir, they are joining.
Okay, okay. Risha, can you hear me? Risha, can you hear me?
Yes, sir.
Okay. You'll be sharing the screen or I have to share the screen.
Sir, I can share.
Can share? Okay.
Yeah.
Then I will make you host. Okay.
Okay, sir.
Let them join. Okay. Then they are
Mm.
joining. Then we can start. Okay. We'll be joining. We'll be starting now. Okay. Risha, can you hear me?
Yes, sir.
Okay. So for today's seminar, Risha Debarma will be speaking. Okay.
Sir, can
So
you share now?
yeah, she'll be talking about anaerobic bacteria. Okay. Yeah, I'll just make you host. Yes, you can do it now.
Mm.
Okay.
Sir, is it visible?
For me, it's visible. Others, can you see the screen?
Yes, sir.
Okay. Can you hear Risha?
Yes, sir.
Yeah. Okay. You can start, Risha.
Yes, sir. Good morning, everyone. So my today's topic is on anaerobic bacteria, Clostridium. The contents are introduction, morphology, cultural characteristics, pathogenicity, and four species of Clostridium, which is Clostridium perfringens, Clostridium tetani, Clostridium botulinum, Clostridium difficile. And so Clostridium is basically gram positive, anaerobic, which is where oxygen acts as a poison for their growth, and it is a spore-forming bacilli. The Clostridium contains bacteria responsible for gas gangrene, food poisoning, tetanus, and botulism. Some of the pathogens, for example, Clostridium perfringens and Clostridium tetani, are normally in human and animal intestine. Morphology. Clostridia are highly plebiomorphic, usually 3 to 8 into 0.4 to 1.2 micrometer in size. Clostridium are motile with peritracheate flagella, with few exceptions such as Clostridium perfringens and Clostridium tetani type 6, which are non-motile. Clostridium perfringens and Clostridium butyricum are capsulated, while others are not. Uh, the spores. Spores are wider than the bacillary bodies, giving the bacillus a swollen appearance resembling a spindle. The spores may be central or equatorial, giving the bacillus a spindle shape, which is in Clostridium bifermentans. Subterminal, where the bacillus appears like a club shape, and which is found in Clostridium perfringens. And the oval and terminal, which resembles like a tennis racket, which is found in Clostridium tertium, and the spherical and terminal, giving a drumstick appearance, which is found in Clostridium tetani. Resistance in spores. The spores exhibit a pronounced but variable resistance to heat, drying, and disinfectants. The vegetative cells of Clostridia do not differ from those of non-sporing bacilli in their resistance to physical and chemical agents. Spores of Clostridium botulinum survive boiling after 3 to 4 hours and even at 105 degrees Celsius, where they are not killed completely in less than 100 minutes. Clostridium tetani spores persist for years in dried earth. All species are killed by autoclaving at 121 degrees Celsius with 20 minutes. The cultural characteristics. Clostridia are anaerobic, uh, where the sensitivity to oxygen varies in different species. Clostridium novae are exacting anaerobes and die on exposure to oxygen, while others like Clostridium histolyticum are tolerant and may even grow aerobically. The optimum temperature for pathogenic Clostridia is 37 degrees Celsius. Some saprophytic Clostridia are thermophilic, while others are psychrophilic. The pathogenicity are exotoxin, where the pathogenic Clostridia form powerful exotoxins. Clostridium botulinum bacilli are virtually non-invasive and non-infectious. Botulism is caused by the ingestion of preformed toxin in food. An invasive, the Clostridium tetani has little invasive property and is confined to the primary site of lodgement. Tetanus results from the action of potent exotoxin it produces. An invasive exotoxin, Clostridium perfringens, besides being toxigenic, is also invasive and can spread tissue and may even cause septicemia. Uh, Clostridium perfringens. Clostridium perfringens is a normal inhabitant of the large intestine of humans and animals. It is found in the feces and contaminates the skins of perineum, buttocks, and thighs. So, when these Clostridium perfringens, they grow in deep compact soil, and when they feel the stress of fresh oxygenated air, they often produce spores which are extremely resilient to the environment and can even survive the cooking. Uh, It typically infects food when it is prepared in large quantities or kept warm for prolonged period. It is found in the environment and contaminates food when they are left for a while. Clostridium perfringens may also get into the wounds when the wounds are made by dirty, sharp objects, which is in contact with soil rich with Clostridium perfringens and other anaerobes. In case of deep wounds, it may cause Clostridium myonecrosis. The spores of Clostridium perfringens are usually destroyed within 5 minutes by boiling. The pathogenicity, it includes toxins where there are four major toxins, that is alpha, beta, epsilon, and iota. The major toxins where the alpha toxin is produced by all types of Clostridium perfringens and most abundantly by type S strains. The beta, epsilon, and iota toxins have lethal and necrotizing properties. The minor toxins, which includes gamma and eta toxin, have minor lethal action, whereas the delta toxin has lethal effect and is hemolytic for the red cells of even-toed ungulates like sheep, pigs, etc. The theta is an oxygen-labile hemolysin antigenically related to streptolysin O. The clinical manifestations of Clostridium perfringens are uh, gas gangrene, which is caused by type A Clostridium perfringens, the food poisoning, which is also caused by type A Clostridium perfringens, and the gas gangrene, which is caused by type D as well as type A Clostridium perfringens, necrotizing enteritis, which is caused by type C strain, and biliary tract infection, brain abscesses, and meningitis, and thoracic infection, which may even follow penetrating wounds of the thorax. Clostridium tetani. It is caused by a causative organism of tetanus, which is widely distributed in soil and in the intestines of humans and animals. It is gram-positive, slender, bacillus, about 4 to 8 to 0.5 micrometer in size, which is Clostridium tetani are not destroyed by 5% phenol, or 0.5% mercury chloride solution in 2 weeks or more. So, like Clostridium perfringens, it also grows in a compact soil, and when the environmental conditions improve, the spores are able to sprout into fully fledged Clostridia. When doing gram stain, Clostridium tetani stains purple or gram-positive, and it is a bacillus, which means it looks like a big cylinder or rod under the microscope. So the pathogenicity. Wash spores injected into experimental animals do not germinate and are destroyed by phagocytes. Germination and toxin production occur only if favorable conditions exist, such as reduced OR potential, devitalized tissues, foreign bodies, or concurrent infection. The toxin produced locally is absorbed by the motor nerve endings and transported to the central nervous system's intra-axonally. The toxins produce at least two distinct, uh, that is hemolysin, also known as titanolysin, which is heat labile, oxygen labile hemolysin, antigenically related to the oxygen labile hemolysin of Clostridium perfringens, and neurotoxin, which is also known as titanospasmin, which disrupt the neuromuscular systems of normals. The laboratory diagnosis of tetanus is usually made on clinical grounds. Clostridium botulinum, which also causes botulism. It is a motile biperitrachic flagella producing subterminal oval and bulging spores. it is all the spores are heat resistance and oxygen resistance, which survives for several hours at 100 degrees Celsius for up to 10 minutes at 120 degrees Celsius. Botulinum is of 8 types. There is A, B, C1, C2, D, E, and F and G. The clinical types which causes foodborne botulism, uh, it is usually caused by a preformed toxin, and the symptoms usually begin 12 to 36 hours after ingestion of food. And wound botulism, which is a very rare condition resulting from wound infection with Clostridium botulinum. Toxin is produced at the site of infection and is absorbed. The symptoms are similar to foodborne botulism, except the gastrointestinal components which are absent. And the infant botulism, it is the toxico infection. Clostridium botulinum spores are ingested in food, which gets established in the gut and produce the toxin. The laboratory diagnosis, it is confirmed by demonstration of the bacillus or the toxin in food or feces. The cultural characteristics, uh, optimum temperature is 35 degrees Celsius, but some strains may grow even at 1 to 5 degrees Celsius. Spores are produced consistently when grown in alkaline glucose gelatin media at 20 to 25 degrees Celsius. The prevention for Clostridium botulinum is it can be achieved by proper canning and preservation, or a prophylactic dose of antitoxin should be given. And Clostridium difficile, which is a long, slender gram-positive bacillus with a pronounced tendency to lose its gram reaction. It is non-hemolytic, saccharolytic, and weakly proteolytic. It is an opportunistic organism that rarely causes disease and does so only when normal flora is lost. Clostridium difficile causes acute colitis, like with blood or watery diarrhea, and pseudomembranous colitis. The pathogenesis, where two high molecular weight exotoxins, that is A and B, are involved in the pathogenesis of the conditions. Toxin A is a potent enterotoxin, and toxin B is a cytotoxin. The diagnosis, which can be done by direct examination of colon by endoscopy to look for microabscesses, or culture on the selective media, detection of toxin A or B in stool by ELISA is the mainstay of diagnosis, demonstrating the toxin in the feces of patient by its characteristic effect on hepatitis 2 or human diploid cell cultures. The toxin is specifically neutralized by Clostridium sordelli antitoxin, and the molecular methods to detect genes for A, toxin A or B. The treatment can be done uh, if the disease is prevented by restricting the use of antibiotics with Clostridium difficile outbreaks, or if the condition is treated by discontinuing the antibiotic causing the disease and insulting the vancomycin or metronidazole. So the conclusion is that Clostridium difficile infection is a major cause of healthcare-related diarrhea, leading to increased morbidity and mortality in surgical patients. Increase in failure rates and resistance to current treatments are clinical economics challenges in the healthcare situation. So my references are uh, Anand Narayan and Panikar's textbook on microbiome. And this is all for my presentation. Thank you.
Okay. Risha, can you hear me?
Yes, sir.
Okay. Give me the total conclusion from your presentation. What was that?
Sir, yes,
Tell
sir.
me. Tell me about the bacteria. About the Clostridium, you tell me. What type of
Yeah,
disease they? What type of symptom you can see in case of Clostridium infection? What type of disease they can give you and transmission? Tell me.
sir, Clostridium, it is anaerobic,
Okay.
which, uh, which is where the oxygen is toxic to them, and they are unable to repair the infection which is caused by oxygen.
Okay. Can it affect human?
Yes, sir.
Yes. Tell me the infection.
Sir, it causes like bending of bodies,
Okay.
like where the head and where the body acts like a arc, and the head and toes are more towards the side of downside.
Okay. Then.
Uh, it also infects the brain.
Okay.
And
What are the symptoms?
symptoms, it causes like fever or drooling and
Diarrhea.
diarrhea, and it also causes irregular urination.
Okay. Tell me the transmission.
Sir, in case of when there's wound,
Okay.
uh, when the wounds are deep, like which is called where the wounds are cut by dirty or sharp objects,
Okay.
and when the the objects are in contact with the soil, which may also contain Clostridium perfringens.
Okay.
So it causes Clostridium myonecrosis.
Okay. Hmm. In your slides, you have mentioned some word like proteolytic. What is that?
Like,
You have mentioned one word, proteolytic. Yes or no?
yes, sir.
Yes. Tell me. What are proteolytic bacteria? Hmm? Tell me, Isha. Can someone tell me what is proteolytic bacteria?
Sir, those bacteria which break down, protein to polypeptide or amino acids.
Protein to polypeptide. Protein and
Yes,
polypeptide
sir.
are same. Proteins and
Sir,
polypeptides are
amino,
same. Mm-hmm.
sir, smaller groups, that is amino acids, two smaller groups.
Those bacteria what? Tell me.
So break down the protein to amino acids.
Okay. For breaking, they need something, no? Those bacteria which
Yes,
produce
sir.
protease enzyme, which can break down
Yes, sir.
protein. Okay. Or they break protein means they break peptide bond. Okay. Risha, tell me about hemolysis
Yes, sir.
or hemolytic. You have mentioned the word. What is that?
Sir, it is uh, where mm,
Mm-hmm. Hemolytic bacteria. You have mentioned in their last classification.
yes, sir.
Yes.
Mm,
Tell
sir,
me.
which where it is destroyed by heat,
Uh-huh. Disrupt by
heat,
heat.
and no, sir, no.
No, not at all. Can anyone tell me about hemolytic bacteria? Yes. Hemolytic bacteria.
Sir, the bacteria that breaks the blood cells, the RBCs.
Okay, very good. Which are grown in blood. Blood agar you used to use, no? Some of the bacteria.
Yes,
For
sir.
growing some bacteria, you use blood agar, no?
Mm.
Yes or
Yes,
no?
sir.
All those are hemolytic bacteria which break down all the red blood cells. Okay, means they induce hemolysis. Okay. If you are having any question to Risha, you can ask. If you are having any question to Risha, you can ask. Are you having any question about this one to Risha? Okay. Risha, what's your roll number?
Sir, roll number 6.
Who is roll number 7?
Sir, there's no roll number 7.
8. 8 is there?
Sir, me.
8. And
Yes.
9? 9?
Sir, Sunena.
Okay. Tomorrow you will be presenting.
Okay, sir.
Roll number 8 will be presenting on tomorrow. Okay.
Okay, sir.
And Sunena is here.
Yes, sir.
You be prepared.
Yes, sir.
If we get time, then we will take your presentation on tomorrow. Okay.
Okay, sir.
Okay. So by evening, by today evening, you just send me the your slides. Okay.
Okay, sir.
Okay. We will just wind up this particular session here only. Okay. Take care of you.